Thank you all for coming today. We're very excited to see so many attendees for our mostly monthly Resilient Speaker Series. First, I want to say, since I know many of you will ask, that we will provide a copy of the slides in the chat. Also, if you would like a certificate of attendance, you will find a link to that once you complete the evaluation. We will provide a link to the evaluation toward the end of today's program. Also, please go ahead and introduce yourself in the chat and feel free to add any questions you may have in the chat. My name is Wallace Dixon, and I serve as the founding director of the ETSU Strong Brain Institute. Now in its sixth year, the Strong Brain Institute works to apply the science of childhood adversity to prevent and mitigate the negative impacts of adverse childhood experiences on both children and adults. We also strive to promote awareness and adoption of resilience-informed practices and policies across ETSU and throughout the Appalachian Highlands region. One of the ways that we advance these objectives is through information dissemination, most notably via our Speaker Series platform. Today, we're proud to present to you our 35th Speaker Series presentation since its inception. Today, I'm honored to introduce Dr. Mary Jo Hedrick, or as we call her around here, MJ. Dr. Hedrick is a research assistant professor in the ETSU Ballad Health Strong Brain Institute and a scholar whose work bridges trauma science, community-based research, and maternal infant health. Her research focuses on how adversity and resilience shape developmental outcomes, with a particular emphasis on rural Appalachian communities. She leads multiple regional and national initiatives translating trauma-informed science into practice across schools, healthcare systems, and family-serving organizations. And so with that, it is my pleasure to turn the platform over to MJ for today's presentation, Epigenetics 101, how experiences get under the skin.
Thank you so much for the introduction, Wally. I really appreciate it. And thank you all so much for being here today. I am so excited to talk about epigenetics, which is one of my absolute favorite topics when we talk about developmental psychology. Hopefully today we're going to talk a little bit about some intro to biology things. We're going to think about high school again just for a little bit. Some pretty foundational things that we see in biology, and then we're going to build onto this epigenetics element. And so a little disclaimer at the very beginning is that epigenetics has a whole bunch of different roles in our development. Today, I'm going to be specifically talking about the way we have studied and understood epigenetics from a developmental psychology perspective and from a perspective of adverse childhood experiences and how experiences shape brain development in particular. There are people who do epigenetic research with cancer patients and epigenetic research with normal, healthy individuals. So there's a lot of different ways that epigenetics can be applied. But specifically today, we're just going to be talking about this kind of adverse childhood experiences, trauma, and brain development element. So with that, let's go ahead and get started. Oh, and the minute I say that, okay, there we go. So the objectives that we have today is I'm going to do a brief refresher of DNA and gene expression. So we're just going to, again, go back to high school or elementary school or middle school whenever we learned about cell duplication and all that fun stuff. So we're going to do a brief overview of DNA and what that looks like. And then we're going to define epigenetics and some key foundational research that we have found in this area in the last couple of decades. We are also going to talk about adverse childhood experiences and how stress and adversity impact the epigenetic expression. Then we're going to get into some really specific studies, some that are old and foundational, and then some that are new that literally came out just this year. And I'm so excited to share those with y'all. And then we're going to discuss a really groundbreaking study that recently just came out about the intergenerational impact of trauma. Then we'll talk a little bit about the way we use that and how this can apply to your work. So first and foremost, hopefully y'all recognize what's on the screen here. So here we have a chromosome and DNA, and then the small part of the strand of DNA is our gene. So again, the gene is just like a line, it's like a message that tells the body how to make proteins and what your body is supposed to be. So one section of a gene may say, hey, we're going to make a kidney now. And one section of a gene may be like, oh, we're going to be the brain, right? So the gene is what holds the information that builds our proteins and our bodies. And the gene is within DNA. And so DNA, it's just storing and transmitting genetic information across growth and development. And we never stop replicating genes and DNA. That's something that happens our entire lives. And then that's all kind of held in a chromosome. And so that's where these DNA strands are wrapped up really tight. And we're going to talk about the way that they're wrapped up together because that's really important for epigenetics. And the way that our DNA is wrapped forms the shapes of these chromosomes here. And at the end of each chromosome is a telomeres. And we will talk about that a little bit as we get into today as well. Okay, so what is a genome? So a genome is basically our complete genetic material in any organism. So we have genomes, other mammals and reptiles, they all have genomes. And so this is basically within every single one of our cells, inside of every single cell, whether it's in the brain or in our stomach or in our foot, we have the same genetic material. And so that's held in the nucleus of our cell. And then we see here how it kind of expands out. The reason I wanted to share this is because cell division and the way we look at a cell and the way the nucleus divides is very important for the way we talk about epigenetics. So that takes me to the kind of central dogma of DNA. And so that is basically the fundamental process in which DNA is used to make proteins. So the function of DNA is to tell our bodies how to build the structures that we need to survive. So when we are in prenatal development, if we're talking about fetal development, our DNA rapidly divides, and each section of our DNA tells the rest of our cells that we need to develop our eyes and we need to develop our legs and things like that. Rapid multiplication, and that's stored within each one of our cells. And in order to do that, there are two main steps to kind of reading our DNA. And so the main one that we need to remember today, if you guys want to call back, is going to be transcription. So transcription is basically when, and I have a little graphic here if you can see this. So transcription is basically where this RNA polymerase is going to, and I'm sure you guys have seen these little videos online of like these little kind of, they look like amoebas walking along a strand of DNA or kind of sliding along the outside. But it basically writes down the message that is within our DNA. And it slides up and down our DNA and basically creates something that we use for translation. So that's when we have our RNA. So that's our messenger RNA. Our RNA goes and tells other parts of our cells to assemble amino acids and proteins, and we need to build a new neuron or we need to build a new kidney, right? And so this transcription, so where this polymerase can move up and down the strand is a very important element in epigenetics, right? The ability to read your epigenome and the ability to read your genome are extremely important. And we'll talk about this a little bit. I'm not going to talk about a lot about translation in this. So like the way that our DNA is turned into other proteins. I'm mostly going to talk about the way that it is transcripted. So the messages that we get from our DNA and how we're able to do that. And then we're going to talk briefly about the cell. Okay, so this is the last little bit of introductory biology that we need to cover before we dive into the fun epigenetic stuff. So we have a cell cycle, and as you can see on my screen, I have like the 4 different phases of the cell, right? But there are 2 main parts. So interphase, that's basically the time where if you see up in this corner over here, that's basically where our chromosomes unravel and we basically have that little messenger that goes up and down the strands of our DNA and makes a duplicate, makes a copy of what's on our DNA. And it gets all messy and it's unwound, but they're duplicating, right? And then in the next phase, so the M phase of the cell cycle, so that's mitosis, right? We have cell division. We now have perfect replicas of the DNA that was in the original cell. Then those start to cleave apart, and then we have two new cells. And so basically, this interphase, like the part where we're copying all the DNA that was in there before, is really where our DNA transcription, translation, replication take place. And that is the phase of cell replication where epigenetics has its biggest impact. And so this will come up. I'll kind of refer to this a couple of times as we go throughout today. And so that leads me to kind of digging into what is epigenetics. So epigenetics is very simply just a change in gene expression without changing the actual DNA itself. I like to think of it as a dimmer switch for a light bulb, which is kind of why I have my light bulb on here. So epi just means above. So it sits on top of the DNA, and I'll show you some pictures of that here in a little bit. And basically what epigenetics does is it turns on and off parts of our DNA strands. So maybe when we're older, we don't need to create, like once we've stopped our kind of preliminary development in early childhood, we're not growing taller anymore. We can stop the epigenetics kind of hops onto the DNA and says, okay, you can stop making new organs, or okay, we can stop making new bones, things like that. And so the epigenetics really just is like a light bulb. We're turning things off and on, we're dimming things, we're making things brighter. And epigenetics is entirely dependent upon the environment. And there's some like innate things, but for the majority, our body adjusts to whatever environment we're in. And so this happens throughout our development, and it's really designed to help us survive and thrive. And so, why does this matter? Epigenetics really helps us understand at a biological level the way our bodies adjust to our experiences. And it really helps understand the link between specifically early childhood trauma. So when we have a lot of growth in our body, a lot of freedom in our brain to connect the way it should, and a lot of new cells and things like that. Our environment shapes the way the epigenome responds to that environment. And so epigenetics really explains how early childhood experiences and repeated stress literally change the function of the systems that we really depend on. So epigenetics changes our stress and hormone regulation. It changes our immune response and immune pathways. It changes brain development, metabolism. And it really gives us a biological link between experiences and long-term health. And it's also really important that we understand epigenetics is bidirectional. Just as adversity can shape gene expression can change epigenetics. So there's like this feedback loop. And the way that this was originally discovered was through twin studies. So I don't know how many of you guys in the room right now are twins, but I'm sure that when you were younger, you and your twin looked pretty similar. You might have had like similar tastes, but as you've gotten older, your experiences literally change the way your DNA expresses. So you may look different as you get older, or you may have different behaviors. So an example I have up here is from a longitudinal data study where we have a quote unquote normal twin and then an affected twin. So at some point in development, this twin had some kind of maybe a traumatic experience. Specifically, this study was done on people who had experienced poverty and neglect. So this twin in particular experienced it at an acute degree to the point where the epigenome changed and it expressed or allowed to express the genes needed for a psychotic episode. So it changed the way serotonin and dopamine were able to function in this person's brain, right? And so that's what individually ultimately changed the way that their DNA expressed. So that's why one twin may have a mental illness and one twin doesn't, or why one twin develops some kind of like skin cancer and one twin doesn't. It's because these experiences change the way our genes express. Even though that twin probably carried the genes needed to express a psychotic episode, that was never expressed because they didn't experience the same duration and intensity of adversity that the other twin did. So that was really the aha moment through twin studies that we figured out people change, right? People change across time. And it's not just something that happens externally, but actually at the level of our gene that is in every single cell. Okay, so what are the key mechanisms of epigenetics? And I'm going to go more in depth into each of these as we go along. So this is a lot of words, and don't worry, I will expand a little bit more. But there are kind of four main ways that epigenetics change the way our genes express. So the most common and the most researched is DNA methylation. And I'll be talking about that a little bit in the next portion. And then we have histone modification. So histones are, I will also explain this later, but they are these kind of units that create the shape of our chromosome. They're like kind of the carby backbone that makes our genes take a specific shape. And then we also see that in emerging research, this has been very, very recent, that we've looked at non-coding RNA. So basically, this happens during translation. I'm not going to cover that right now, where basically there's an issue in the messenger or chromatin structure changes. And that would be at the level of the structure as well. But that's very, very new. I'm going to go with our classic epigenetic understanding, especially when we talk about early childhood experiences. And so, like I said earlier, just a little caveat. This is a very controversial field. Lots of people study epigenetics and have multiple perspectives on the way that it functions. So what I'm going to do is do my absolute best to translate the foundations and then some emerging research that we see in epigenetics. And really, I'm going to talk about the things that have the most evidence base. And I have a whole stack of peer-reviewed journal articles sitting right here on my desk that I have read over and refreshed myself on if you guys have additional questions about the way this may look. And I'll be taking questions at the end. Okay, so let's talk about methylation. Again, we're going to go a little bit into early biology here. So DNA methylation is very simple. A methyl group is attached to the 5' position of cytosine, causing gene silencing in 2 ways, okay? And I'll show you a fun little graphic that helps me for this. So basically, adding a methyl group interferes with transcription. So like I talked about earlier, we have that little RNA moving back and forth, trying to copy what is in our DNA. Adding that little cytosine, so we add a methyl group on top of this, we get stuck, right? The RNA translator basically gets stuck somewhere along the way and can't move forward. And then we have this kind of methylation that's also reinforced through specific histones. And I'll talk about that here in just a second. So this is my favorite picture. And if you guys, I have a QR code that is at the very end of my slides. It has some references, but this is from a YouTube channel that does like really funny drawings of biology. And as I was searching, I found these amazing presentations. And so I'll go ahead and talk a little bit about what we're seeing here. So along our strand here, we see that there's an additional methyl group added, and that would be on our cytosine, right? With this additional methyl group here and here, it has turned off gene expression. And that means we cannot transcribe what is in this section of DNA, right? So that can be a lot of different things. So we can't have our nice polymerase, our RNA, move up and down this because our methyl group is blocking it. However, when our little methyl group floats off, no, I can't attach anymore, and they're not attached down here, we are able to transcribe and translate what is happening in this methyl group. And so these are really kind of the methyl groups add as kind of like little roadblocks for our RNA translator to move through. And so in this case, when something is methylated, that means we are not translating. When something is demethylated, that means it is expressing. Okay, so that will come up as we talk a little bit more about our studies. Okay, so what is histone modification? So histone modification, like I said earlier, is when it is what makes up the structure of our chromosomes, right? So it's what helps us keep that like little wishbone shape on either side of our histones. So histones are basically just what our DNA wraps around. And basically adding a methyl group or some kind of messenger cell, as you see this animation, there's like a little red dot coming through. Adding a methyl group or another type of chemical messenger to a histone will cause them to contract and tighten up. And that really determines whether or not an entire strand of protein gets translated or transcripted. And so I really like this. Again, I love these little cartoons that we have here. So everyone just hang loose, ride that transcription wave. These guys are loose, they're free flowing, they're chill, and all of the DNA that's wrapped around our histones here can be replicated, right? So anything that we learn there, anything that our body needs, it's easily accessible. But when they get all uptight, our like little pieced out group says, I'm out. I'm going to actually have y'all tighten up here, and we're not going to be able to translate or transcribe the DNA that is in this cluster here. And something that's really important about histone modification is one of the few changes at the epigenetic level that is genetically heritable. So specifically, and I'll talk about this in more detail here in a little bit, specifically the way our histones are shaped in both the paternal and maternal DNA will shape the way an infant's DNA is expressed. So this is something that survives all of the kind of clean slate replication of early fetal development. And it's stable through all cell division. So even if your cell is dividing rapidly, the histone modification will stay the same. However, it can be reversed through a demethylase, which I'm not going to get into the weeds on that. But all you need to know is that reversing histone modification is very hard. And there are very few times where that happens all the way through. Okay, so what is some animal behavior? How did we figure all of this stuff out, and how does it apply practically? So, my favorite study in epigenetics, and this is the thing that completely changed my life when I began to learn about developmental psychology, was an early study by Michael Meany and Moish Sivs. They are they're more like pharmacologists that kind of stumbled upon this work. And so they conducted a rat study where they had two groups of female rats. All of them were pregnant. One group of female rats were stressed out. They were in an environment where they didn't have a lot of things to make their bed. They weren't able to nest the way they wanted to. And there were pheromones of potential predators that were released in the air around where they were. And then we had another group of moms in the same group of mother rats that were given an enriched environment and all the things that they needed to thrive. And they were going out living their best life and being social and having a really great time. And what happened is after those rat moms had their little babies, the mothers who weren't stressed out were loving up on their babies. Specifically, they were licking them a lot. And that is the equivalent of almost like this serve and return behavior that we see between mamas and babies now, where they were attentive to their needs and they were licking them and they were having a good old time, right? However, the stressed out mama pups, their babies didn't get licked a lot because mom was in survival mode. Like she didn't have the right bedding. She smelled a predator. So she was a little bit more distant. She wasn't as okay with the licking and grooming behaviors. And so what we found when the researchers looked at their DNA and the things that were being expressed is they found that maternal behavior actually changed the methylation of these rats. And so I have this great picture here. So when a mother was licking and grooming, this specific line of DNA, and I'll talk about which one it is here in a little bit, it was their NRC3 1 strand, which is basically our serotonin receptors. It's what tells us to kind of leave serotonin in the synaptic cleft and to keep getting more serotonin. It was turned on. So that means that the baby pups felt calm and confident and loved, and they were able to go through a maze like nobody's problem. They could go and find their Fruit Loops, which was actually really what they did with this study, is they reinforced the rats with Fruit Loops. But a mom who was really scared and didn't lick on her babies a lot, they found these huge methyl groups attached to the strand of their DNA that was responsible for the encoding of serotonin receptors or serotonin transporters. And so what they found is that these kids were already getting ready for a stressful life. They weren't able to kind of go through a maze as quickly. They weren't as sociable. They had a hard time regulating their own stress response. Even though the maternal stress impacted their caregiving, they were not in a stressful environment. They were in the same environment as the other rats. But they were still anxious and they didn't socialize and they had a really hard time. They were stuck in that survival mode. And this is really the foundation of what we understand as the HPA axis overactivation in our kiddos. So this translates very directly to humans, and we've been able to replicate it through a lot of different studies. And this maps directly onto the work that was done in the 90s with adverse childhood experiences. And so we see a dose response relationship with ACEs. And so basically, the more adverse childhood experiences, the more abuse, neglect, household dysfunction that you have, the more likely you are to have depression, anxiety, substance use, diabetes, heart disease, autoimmune disorders. And for decades since the Meany and Sieves article, we have found a direct link between epigenetic changes, like the way our rat moms treated their babies early on, preparing them for the environment, with what we see in ACEs. And so this doesn't just happen to a child. This isn't just like a really hard experience. It is a really hard experience, but it changes them at the level of their genome. It literally changes the ability of their body to produce a serotonin transporter, which keeps us happier and keeps us healthier. And so what we really talk about now is the biological embedding of adverse childhood experiences. So the way that this actually becomes a part of our DNA. So that really comes down to, again, I'm going to return back to that Meany and Sieves article, which is also linked in my resources if you guys want to read it yourselves. What they found is that in the highly stressed out rats that didn't have a lot of maternal caregiving, we found they had an overactive HPA axis that was responding constantly. So there was a really high cortisol release. And in a healthy system, we would expect cortisol to release, it does its job, and then the negative feedback loop turns off, right? So we're now out of danger. I got that spike of cortisol to run past that scary bear, and now I'm good. The problem with the exposed rats is that that system never turned off. They continued getting that cortisol spike and that stressful response throughout their development, throughout their life. And so we often see that epigenetic changes happen to the specific parts of the genes that regulate cortisol, that control inflammation in our stress response. And they're all involved in emotional regulation. And ultimately, it's really important to understand these epigenetic changes, especially in light of this model that we've been using for probably a decade now about adverse childhood experiences, where we have this generational and historical trauma, social conditions, adverse childhood experiences that leads to actual changes at the level of our DNA in the people who have experienced these ACEs. And the HPA axis activation and the increased cortisol, that also has a lot to do with increased inflammation and chronic stress and things like that, which ultimately lead to disease and early death. So we do see these kind of long-term effects. Okay, so what are these genes that are changed by epigenetics? So these are not by any means a comprehensive list of exactly every single thing that's changed as a result of epigenetic changes to adversity. You can see my little rat mom and her baby here. These are specifically the genes that we have found replicated dozens of times at this point in both rodent and human models. Okay, so the first gene that I want to talk about is the NR3C1 gene. And so this is our glucocorticoid receptor gene. Sorry, I had to write that down, like the phonetic spelling of that. And this is really what helps us regulate cortisol. And there's so much nuance around cortisol. And I know if Andy's on this, she'll probably have a lot to say about cortisol and its ability to shape development. But what we see is that there are increased methylation of this part of the gene that codes for our glucocorticoid receptor. There's increased methylation after stress, which ultimately leads to heightened stress sensitivity and stress reactivity in infants and adults. The next gene I want to talk about is the FKBP5. And this is really a part of our gene that is also related to cortisol. And we see this most strongly in people who have diagnoses of PTSD. So we're thinking about really chronic and persistent exposure to trauma. And we've seen this both in childhood and in adult studies. And it is one of the best replicated genetic markers that we've seen across all the studies that I'll discuss here in a little bit. The next one, and the one that has some really emerging evidence in the last decade, is our BDNF, which is actually our brain-derived neurotrophic growth factor. And so basically, that part of the gene is what's related to our neuroplasticity, or our ability to learn different things and have some flexibility in what we learn. And so basically, the gene is methylated, so it's not expressed that we have this growth, and it ultimately leads to less overall neuronal growth, right? So that can sometimes be related to the lower volume that we see of people who have high trauma backgrounds in their gray and white matter of their brain. And so this BDNF typically supports neuroplasticity, and we see an overall decrease in expression after experiences of trauma and adversity. And then this final gene that we'll talk about here, this is the SLC6A4 gene. It's the serotonin transporter gene that I was talking about earlier with our mama and baby rats. This is really responsible for controlling our serotonin system. So the things that help us with rewards and maintaining mood and affect, it is absolutely critical for mood regulation. And it's also extremely sensitive based on the research that we have today to maternal stress exposure. And something I want to note, these are not bad genes. I want to emphasize that these are all here to help us survive and thrive, and they're adaptive. They are what help us be prepared for our next environment. And something I want to keep in mind is I don't want to at all demonize like people who might be in a heightened stress response, or I don't want to demonize epigenetics, right? We know that our bodies are taking great care of us. And our brains knew when this little baby rat came into the world and the mom wasn't licking them and giving them the care that they needed, the body's like, okay, I got this. I can help us prepare. We can be on high alert. We can be safe. We'll keep ourselves safe. And if we really think about that is occurring at the level of our epigenome, these changes are so embedded in the structure of our own bodies. And basically, when we have this kind of uncertainty in our built environments and in our childhood, we see these as long-lasting changes. So I've talked a good bit about rats and the way we've kind of figured this out with them. And so now I'm going to kind of move on to a little bit more of our human evidence, specifically around childhood trauma. So how do these patterns really kind of show up in our humans and children and adults? So one of the earliest, most influential studies was conducted by Patrick McGowan and colleagues who examined specifically the hippocampus. So that's kind of a central structure in our brain. We generally think of the hippocampus as a place where memories are stored or directions and things like that. It serves a lot of different purposes. But what McGowan and his colleagues did is they actually examined post-mortem the hippocampal tissue of people who had committed suicide and had a documented history of chronic abuse, right? So a very heavy, dark topic. But what they found mirrored exactly what Meany and Sieves found in their studies. They found that there was higher methylation of the NRC31 transporter. So that means the people who ended up having this childhood trauma and committing suicide, they were not able to regulate stress as well on a level of their DNA. We also saw a lower expression of the glucosteroid receptor. I cannot say that word. We also saw decreased, and so that means there was decreased cortisol regulation. And then basically, they had enough evidence to really say that these two genes, so the NR3C1 and the FKBP5, are what we now call, and the NRC31, I have that here, they are actually biomarkers now that we look for in research for people who have dealt with chronic stress. And so this was really a pivotal turning point in our field and understanding the impacts of early adversity and early trauma that literally changed the way our brains build themselves and the way that we are built around our environment. And so since that study, this has been replicated time and time again through a variety of means using like umbilical cord blood, saliva, and additional blood findings from healthy adults, not from umbilical cords. And his was the first, so to kind of tie that back in, McGowan was the first person to ever look at the actual structure of the brain and look at the level of the gene. And so now we've replicated that time and time again. And again, across dozens of studies, more than I could ever count, we have seen these chronic changes at the level of our DNA. And one of those, and one of the most exciting projects that has come out of an historical event, is the Dutch famine study. And so basically the Dutch famine study, and I can talk, the Dutch winter famine study is a natural experiment. And this happened, oh my goodness, the dates are leaving my brain right now. But basically there was a famine and there were entire generations of families that were impacted by this, even though it spanned a good 10-year period. And basically what they found in doing this giant longitudinal data analysis, they were trying to understand why people who were exposed in the famine and who weren't exposed in the famine were having different health outcomes, even after they had been given appropriate food and were being healthy. What they found is that because of the stress that occurred during this famine and the way that their brain structures changed during this period of scarcity, it was linked to increased biological aging. So these adverse experiences, specifically in this case, it would be like a giant historical event where they chronically went without food. Their body attuned to what was going on and was like, okay, we need to release cortisol. This is a stressful situation. We need to be aware and alert and understanding of where our next meal is coming from. And basically, what they found is that the people who had experienced that as an adverse event had increased biological age. That means that their bodies were older than their actual age was. So if you were 25, your DNA says, oh no, you're actually 50, right? And that leads to some of these chronic health outcomes that we saw in that ACEs pyramid, if we're thinking back, and things that we see here. We see that there are like increased heart disease because our body isn't replicating as much new heart tissue to restore any kind of damage. We see increased risks of chronic pain because our body isn't replicating as much of those good feeling drugs that help us get through the pain and adjust, right? And we see this because of this longitudinal data study, that there's a dose response effect. And we saw with all of these folks who had experienced this chronic stressor, they had high rates of cardiometabolic diseases, they had incredible immune dysfunction and more autoimmune disorders, and an overall shorter life expectancy. So again, this isn't just related to you have adverse childhood experiences, you make bad decisions. No, you have adverse childhood experiences. Your body adjusts to the environment that you're in. It puts you in stress. It biologically ages you faster, and you're more susceptible to disease and more chronic illness in general. And so this next, I want to be aware of time here. This next study that I have found absolutely riveting, it was just published this year. And I think this is incredibly important to understand for intergenerational transmission of trauma. So this is more of a proof of concept paper. This is a very controversial article that was just published this year. And basically, they worked with Syrian refugees across 3 generations. And what they did is they followed these generations of Syrian refugees that were experiencing persecution and had to flee their home, right? And if you see my graph over here, in 1980, there was a group that experienced the direct exposure to trauma. So a direct exposure to having to flee their country and a lot of conflict-based trauma. They had a group of women in the 1980s that they followed who were currently pregnant in 1980. And then there was another refugee crisis in 2011, and we found that they also followed a group of pregnant women during that time. And then they had a control group of people who were not exposed to any kind of refugee or stress in that way. And so what they basically found is that for the exposure in the 1980s, the women who were exposed directly, so I also want to note the women who were in this study here, if you see the purple, they were exposed to the trauma directly, right? They were exposed to it early. This was an unexposed population, whereas the next generation was exposed. And then for this, there are 3 generations of unexposed. But here we see that the grandmother was pregnant during this stressful time. She was pregnant. In this case, they followed only women. And they found that at the level of the embryo and the embryos that were developing within the fetus. So if you don't already know this from kind of some foundational biology, is that when a female is developing, she develops in utero with all of the eggs that she will ever have in her entire life. The experiences that her mother has while she is pregnant with that fetus actually changes the way the daughter, in this case, her eggs develop. And so we now follow this baby that is in mom and then becomes like a full-grown adult. And she was not exposed to the trauma, but her baby still carries the same methylation and the same epigenetic changes that her mother did and her grandmother. And then we saw those same changes. Again, this generation and this generation were not exposed to any traumatic events like being a refugee. It was exposed prenatally. And these women had the same epigenetic markers for stress across the germline. And so the germline is basically when you are a gamete cell within a uterus, that is the germline that develops there. And so that's the little itty bitty beginnings of a human. And then we found in the 2011 group that it was more an environmental exposure, and I can talk about that a little bit more. And then we had our control that didn't have any exposure. And I'm going to read this because I think these findings are super, super important. So 21 different regions were methylated across direct exposure to violence. So that means 21 different genes that were all responsible for stress regulation, serotonin production, and many others were silenced across all exposure to violence. That means their ability to regulate stress and regulate cortisol and serotonin were all lessened. So they were not able to handle as much stress. And then 14 regions were methylated. And again, we're going to go back to that histone modification where the histones are wrapped really tightly so the transcription and translation can't take place. 14 different regions were directly just related to the germline exposure. So that means 14 of these modifications only happened at the level of like embryo development. And so, and strikingly, these were consistent across these generations. Like we saw these same methylation in the third generation of children. And so this really was a groundbreaking study. Again, this has just come out this year. The link is in my resources if you want to read it, and all the arguing that's going back and forth about this. They basically found that children with prenatal exposure to violence showed an epigenetic age acceleration, highlighting the sensitive period of in utero development. But maternal exposure was not the whole story. A separate set of studies that was done kind of as a branch of this talked about paternal stress. So this study was almost entirely done with women, and historically we've talked about women being the carriers of epigenetic changes. Well, we actually just have a study, and this is also linked in my resources. There's a new study that's emerged about the way that DNA that is within sperm actually carries epigenetic changes that we have never been able to study before. So we really want to understand the paternal element. Again, this is a new and emerging field, but I don't want us to leave here today thinking, oh, the mom is the only person that's important for healthy embryonic development. It's actually also in the hands with the paternal contributor. And so really, this work here has been absolutely groundbreaking in the way that we understand how adverse experiences can impact and shape generations and generational responses. And we also know that in the nuance of this study that I don't have the time to get into, we know that social transmission, parenting practices, family stress, community context, those are all very essential elements for these findings and what we have found. So there's not just biological transmission, but there's also social transmission. So if we think about the stress of our licking rats from the median sieve study early on, this mom is probably stressed out. Without intervention, she'll also raise her kids to be stressed out. She might not give them the attention and the nurturing that she needs. Same thing with our paternal contributors. And it's never just one thing. It's never just a biological inheritance. It's both. Both have to work together. Our environments are extremely important. And so that leads me to talking a little bit more about the really strong grounded research that we have in maternal infant health. And it's why I'm so passionate about maternal infant health. Our paternal health, we care so much about them, but there's not a lot of research on them quite yet. So I'm going to talk a little bit more about this because there is more converging evidence. So if there's one message I want you guys to walk away here with today, is pregnancy and early infancy are some of the most epigenetically sensitive periods in development. These early experiences, exposure in utero, really affect the entire lifespan. Why? And I think, again, going back to our early biology, we know that fetal cells divide rapidly. Epigenetic changes are being placed, removed, and reorganized based on the need that that kiddo has and the environment that the mom is interacting with. And its stress system is developing to come into a situation that it's prepared for. So I want you guys to kind of put yourselves in the position of a refugee, right? You're a pregnant refugee. Your baby is probably not going to come out into the world and be really successful if they are the most outgoing and exploring and hands-on baby that's happy to cry and talk to anyone, right? It's probably the baby's best bet that they are able to kind of cling, stay small, stay close. If we're thinking about that from almost an evolutionary biology perspective, these are very adaptive and it's calibrating itself to the environment. And so there was this really amazing study that was done by Oberlander, and this is also in my resources. And so he examined women throughout pregnancy, and he basically found that there is, for mothers who had high rates of depression and anxiety during their pregnancy, they found methylated serotonin transport genes in the umbilical cord. So we actually find that just during that window of pregnancy, having stress is something that is immediately provided to the fetus. And we also found that following 3 months after birth, children that had stressed or depressed mothers had higher rates of cortisol compared to a kind of control group. And so that really tells us that maternal health helps tune the child's responses to their environment and bring them in the way that they're going to be the most successful based on the environment that they're in. And some of the most shocking findings that we've been able to find in this work is a lot of maternal care, including like taking vitamins, going for walks, things like that that will de-stress. We've seen that women who are in high neglect abuse situations have babies have higher immune response disorders. So they have immune function genes that are methylated, stress response pathways, and again, that early aging of their body. And so the intergenerational study violence from the refugees that we just talked about found that children with prenatal exposure to violence had an accelerated epigenetic aging even before the age of 10. So kiddos who were even in these earliest stages of their life, their body was always already aging faster. Their health was aging faster. And so from like a public health policy perspective, this really reinforces why the emphasis on prenatal health, women's reproductive research, women's work in general around research is so essential. Reducing stress during pregnancy is not just like nice to have or get it if you can, it's actually going to shape generations. So making sure that we make this a priority is so, so important because it will change the way the next generation of kiddos are able to grow up and have healthy health trajectories. Okay, so I know that this is a lot and this might have been a slightly depressing. However, we do know that there are some ways that we can see changes in epigenetic expression, specifically at the level of our methylation, not as much at the histone modification, based off of interventions that we've developed. So this is really hopeful and actionable, I hope. Epigenetics is not just about this kind of damage or adversity mindset, but it's also about the capacity to change. So epigenetics is always happening. We're always changing. Sometimes, like the histone modification, it takes a little bit more. Methylation is easier to change. And something that we have found is that after frequent and intense intervention, the epigenetic markers can actually change. So specifically, we've seen methylation changes as a result of psychotherapy, nurturing caregiving. So the opposite like of our rat study that we talked about earlier, having stable supportive relationships and adequate nutrition, exercise, and reducing chronic stress all change the way that our DNA methylates after we're through kind of these critical developmental periods. So these changes still can occur later on in life. So this isn't something that's necessarily set in stone. It's something that we can see change throughout life. And so, specifically, there was a study that I've also linked in my resources where they worked with folks who were diagnosed with PTSD. And so, they did a buccal swab. So, you can actually measure because your genes are in every cell of your body. They took buccal cells from the inside of their cheeks and they measured the epigenetic methylation before treatment and after treatment. And they were able to see changes in stress regulation at the level of their gene based on this kind of intense intervention for people with PTSD. Another thing that we have found through buccal swabs, blood tests, things like that, that positive caregiving really changes a lot of this early methylation. So it's one of the consistent findings. It's basically like this sensitive, supportive, stable caregiving reverses a lot of the early methylation that maybe happened in utero. So if you were a stressed out mom, maybe you were having a hard pregnancy, you were having health complications, you had increased anxiety. I think about our mamas who were pregnant during COVID. Like that was just a stressful time. We have found through research that sensitive supportive caregiving reverses that methylation. So the baby comes out into the world, it's ready for a stressful situation. And then we're actually able to see through sensitive supportive caregiving that they adjust back and their DNA, their epigenetic changes. And then social context really does matter. So genes, again, don't operate in isolation. We are changed by our environment. So we think about all the social determinants of health if we're talking about this from like a public health perspective. Things like poverty, racism, neighborhood safety, food access, all influence the stress load that's placed on the body and will ultimately impact the way that our epigenome functions. And so I really want to drive this home. Resilience is not about grit or something that's innate in your personality, right? Resilience is something that can happen at the level of your genome. If you are struggling with stress, if you are overwhelmed, it is not because you are not resilient. It may be because you don't have the receptors available to help moderate some of that stress, right? So I don't want anyone to think that like there's a deficit. You can always build those back up and demethylate. But grit is not about it. I mean, resilience isn't about grit. It's about resources, relationships, safe, supportive caregiving, and environments that are there to help you support adaptation. And so I really want to kind of close out this portion of lots of content with basically the big idea that epigenetics is not determinism. I don't want us to leave here thinking, much like some of the early ACEs work, that ACE is destiny, right? Our epigenetics is not our destiny. It's more of a story of potential and what we're able to do. And explains why early adversity matters, but it also explains why the intervention work that I know so many of you on this call do, like reading to your kiddo, doing interventions in a library, having a safe, stable, supportive teacher. This all reverses the effects of these chronic stressors, this DNA methylation that we see at the epigenetic level. And so, again, biology is not destiny, but our experiences are deeply embedded in our own biology. And so, as exciting as all of this is, I do want to add some caveats. And I'm probably talking really fast. I want to give us plenty of time for questions and conversations. I do want to be very precise about epigenetic limitations. Some of these studies that I've discussed today are correlation, not causation. With the rats, we can, in somewhat good conscience, depending on what the IRB or the Institutional Animal Review Board says, we can't randomly assign humans to stressful situations and then take a sample from them, right? We can do that with rats. That's what a huge part of the research is. So some of the work that we've done, like that longitudinal Syrian refugee study, that is something that was a historical event, and we did have a control group in that study. But at the end of the day, we have so many other variables that may confound those findings. Also, there are tissue differences. So many of the studies that I talked about measure methylation in saliva or blood because we can't sample the human brain, unlike an earlier study that I talked about with McGowan. Um, but they're imperfect proxies. They're not always the perfect picture of exactly what's going on in your DNA. And then lastly, sample sizes can be small. So we don't want to say that this is generalizable for every single person. However, we do have a lot of converging evidence. So we can say with good certainty that this is more than likely what is going on. And the field is still evolving. We're learning new things about epigenetics. I read this amazing article of two epigenetics like researchers in the field arguing back and forth about a very fundamental concept, right? This is evolving, this is changing. And we don't want to use this as a diagnosis, right? Because we took a swab and a person has methylated serotonin transporters, that doesn't mean they're diagnosed with something like PTSD or trauma. And so basically the message is like epigenetics provides really powerful insights, but we have to be hesitant and hedging with the way that we present this. So that kind of leads us um, to like the ethics and misuse. So we want to avoid determinism. There is no one trauma fingerprint. We're not going to be able to have one simple picture. Um, I really want us to kind of turn away from maternal blame. Again, this is all reversible, and a mom is doing the best that she can. And we also want to think about privacy. Obviously, we don't want to bring up potential opportunities for people to say you are genetically inferior because of the way your epigenome has functioned and the stress response that you're in. So again, this is not destiny. This does not mean that if a person has this kind of background or demographic factors that it will ultimately lead to this negative outcome or they will be this specific way. And I really want us to encourage shifting science from individual responsibility. So, mom, why didn't you do this thing differently? To system, how can we support you? How can we decrease the stress? Thinking about universal childcare or things that are early protective factors for our kiddos. So, things like safe community, stable housing, food security, trauma informed care, policies and practices, those all influence the way we're able to address chronic stress. So, what does this mean for you? How can you personally, potentially, reverse epigenetic changes? So, what do we do? The number one thing I want to say is support your pregnant person. Fetal development is so epigenetically sensitive, and supporting maternal mental health, reducing stress, providing wraparound services not only provides a benefit to the pregnant person, but also to the fetus and the person that they're bringing into the world. Another thing that I think is super, super, super important is that we really invest in trauma-informed systems. If we understand that at the level of our DNA, at the level of our genome, our epigenome changes based on our experiences, how can we not change the way we interact with people, right? A person who may seem really dysregulated and disconnected, there are so many other things going on, even at the level of their biology, that may keep them from being able to really connect or regulate. The next thing is early childhood investment. And I will forever say how important this is high quality caregiving, having safe, stable, supportive, nurturing adults. That has evidence at the epigenome that we can reverse these kind of intergenerational effects of trauma through this safe, stable, supportive caregiving. And why I'm such a passionate advocate for this type of work. We can see measurable biological differences based on safe, supportive caregiving. Mental health interventions, again, for those populations that had PTSD, the pre-test, post-test of the DNA that they had methylated, there was a significant change based on this intervention to their ability, their body's ability to regulate genes that regulate stress response. Mental health interventions are an absolute necessity and should be prioritized. I especially think of all of our friends at the VA who work tirelessly with folks who might have PTSD in those backgrounds, and the priority that we should place on these, that they're not just changing something that's happening in a social context, but at a biological level. And then lastly, I always want to do like a call to community and policy action. My favorite thing about this kind of work is that we also have positive childhood experiences, right? So, and a couple of those are like having two safe, stable, supportive, nurturing adults that are in the environment that aren't their parents, that are there for them. That reverses a lot of the epigenetic changes that lead to dysregulation and cortisol. Having a strong, supportive community. These are all things that we can do. Maybe going to like get coffee with a mom who's pregnant with a pregnant person, or participating in things like the Woolly Worm Festival or the fair where kids get to have fun and play and be in a safe environment. These are all things that we can affect change at a large level. And I think I might just skip the case study here because I want time for questions. So some future directions. And this is my favorite meme because I hope that this rings true even after we've gone through all of this. So this is our little methyl group blocking our polymerase that's trying to transcript and translate our DNA. So I died when I laughed at this. You guys might not. But basically some future directions that we see. This field, like I've said before, is expanding and growing, and we're seeing more and more longitudinal studies that follow these kiddos and these folks across time. We also see kind of individual methylation patterns in specialized cells that people are digging into, like specifically the cells that function in our brain and neurotransmitters. We've seen a lot of this work happen around dementia and Alzheimer's and things like that. We see tons of intervention studies using this as a dependent variable or an outcome of intervention that folks are measuring. And then some key takeaways before we kind of launch into questions and resources. So experiences shape biology. That should be one of the big things you come away with. Not just stress and violence, but also safety, love, and support. Not just these hard things that happen, but also the positive things that happen, like the safe, supportive caregiving. Adversity becomes embedded in stress systems, immune pathways, and epigenetic regulation. Adversity is something that really changes the way our bodies respond to the environment. This is not just something that we don't just talk about trauma-informed practices because we really want people to like be nicer. No, I mean, that's one of the reasons, but also we really want people to understand that this is happening at a biological level for people. Healing changes biology. Epigenetic markers are dynamic. Again, safe supportive caregiving can reverse a lot of these methylations that occur. And lastly, love your pregnant people. It's such a sensitive period. We want to support them. We want to love them. We want to give not only them, but their future kiddo the best chance at having a regulated nervous system and being healthy. And last, but not the most importantly, is biology is not destiny. Things can change. And that's, I think, the true story of epigenetics is that our body is this amazing machine that adapts to what we need and helps us overcome so much. So I'm going to open the floor for questions, comments, concerns, and I'm also going to leave this QR code up here and I'll answer some questions. At that QR code, I have like a YouTube playlist and a bunch of citations that I've talked about today. I had like 60 citations that made this today. That's not all of them, but there are some good resources on there. So now I will open it up to questions. You're more than welcome to put something in the chat or come off mute and ask questions. I'll leave this up for just a little bit longer. I'll also make sure if you guys request the slides, this reference is also in the slide so you can scan it and all that good stuff. Any questions?
I'll just make a comment, Mary Jo, while people are thinking of their questions, but really appreciate kind of your framing for us of, when when you start hearing the damage, the biological damage, you do start to get kind of like, oh my gosh, such an uphill battle, but reminding us that of what can shift it and what can change. And it's not a mountain. It is as much as supporting a mom and taking a kid to a park, that all of these little acts actually go towards making the changes. So I appreciate you, kind of your framing that.
Yeah, thank you. I appreciate that. Any other questions? I'm not looking at the chat, so.
People are saying thank you. They appreciate the
Awesome.
information.
Yeah. And like if you guys want any more information about this, I know that like I gave a very high level overview and I had to do some biology primers at the very beginning. But if you're ever interested in anything else, I love this topic so much and I'm happy to chat about it with you and share more resources.
Mary Jo, are you able to comment on how substance abuse plays into epigenetics?
So we talk kind of about that experimental control. We do see that in very similar contexts of people who specifically were substance exposed in utero and are substance using now, we see overall a downregulation of their endogenous opioid system. So I've talked with Andy about that one. But at the level of the genome, we see lower, um, there's a long acronym for it, but it's basically like our self-soothing ability. Basically, we see that it's a lot harder for people to soothe and calm and return to like this centered place. It's not the same cortisol function that I talked about here. It's actually an endogenous opioid and dopaminergic system. They haven't been studied as much as these other genes and these things that I've talked about, but we do see those changes. However, the really great thing is that it is reversible, again, through good relationships is like the most evidence that we have, and intentional intervention. And so that has really, we've seen a lot of that being able to be reversed in the emerging literature that we have now.
Thanks.
Yeah. Any other questions? I was really thinking that y'all were just going to light me up. I have like stacks of papers over here. I have.
Well, you had a really nice style for us who haven't been in biology for a lot of years and have heard the words, but not really sure how they go together. It was a nice way of kind of giving us foundation and being able to understand the impact of some of the research.
Thank you. I'll stop sharing so I can like see people and
Feel free to unmute if you want
yeah,
to ask a question. Sam said from biology to hope.
Oh,
That was a beautiful
that.
synopsis.
Oh, that's great. But yeah, if y'all don't have any questions, like always feel free to like reach out to me. And this is a lot of information. I feel like it takes time to reflect and digest on all of this. But one thing I would love for you guys to share is what is one thing that like stood out to you? Just share it in the chat. Like, what is one thing you'll be walking away with today? Like a word or a phrase, something that stood out to you. Well, I haven't gotten a word or phrase, but support your pregnant people. Yes. Love that. Wally, better understanding of methylation. And Sam, yes, I would love to present to the Ukrainian Psychological Association. I'm sure the time difference will be fun, but we can figure it out. Genetic is plastic, focus on systems, not individuals. I love that. Yeah, intergenerational trauma evidence. Yeah, you're not broken forever. The power of connection. Oh, Jenny, I love that. Oh, you're good at connection anyway, Jenny. So I love that.
And
Yeah.
Chloe said she didn't even know what epigenetics was before this, and now she's happy to know more.
Oh, that's awesome. Reinforcing that change is always possible. Yes, I love that. Mm-hmm. Environment. Yeah. So important. Yeah.
And as a reminder, Lisa had put in the slides, so you will have those and you have the resources. The YouTube video will go up in a few days. And so even if there's a piece of it you want to look at again, feel free to use it, or if using it in any of your own internal trainings, or, the SBI recordings from the Resilience series are there for folks to use in your own work as you're trying to address these big issues that we're talking about, is developing more resilience in our at-risk folks. And we appreciate you all being here today.